Welcome to This Explainer. I am so excited to have you here today because, well, we're essentially going to be lab detectives. We are cracking the biochemical code of a protein's makeup by revisiting a classic and, honestly, a fascinating technique in biochemistry. It's basically a way to see the invisible. So let's step right into the lab and break down exactly how this whole thing works. Okay, let's dive into this. How do we reveal the hidden building blocks of proteins? Take a super simple biological sample, like, say, egg albumin. To us, it just looks like a clear liquid, right? But it's actually hiding this incredibly complex chain of building blocks. The big question is, how do we reveal what's completely invisible to the naked eye? How do we definitively prove what's actually hiding inside that totally plain-looking sample? That brings us to phase one, the protein mystery. Think of it like we're hunting down some very specific suspects that are just laying low, hiding right there inside the protein structure. To catch these suspects, our primary diagnostic tool is going to be the Xanthoproteic Test. Now, this is a highly specialized biochemical test, and it's used specifically to detect the presence of aromatic amino acids hidden in proteins. For today's experiment, we are targeting three very specific amino acids. We're looking for tyrosine, tryptophan, and phenylalanine. If any of those three specific aromatic amino acids are present in our sample, well, the Xanthoproteic Test is exactly how we isolate and identify them. All right, moving on to phase two, the lab setup. I want you to literally picture yourself at the workbench right now, gathering up all your investigative tools. Here is the exact toolkit we need for our detective work. First up, our primary piece of evidence, the protein sample. Like I mentioned, we're using egg albumin for this. Next, we need our chemical reagents. We'll need concentrated nitric acid and then a strong alkali. Now, this can be either sodium hydroxide or ammonia. Either one works. And finally, our staging area. We need some nice clean test tubes and a pipette so we can get those truly accurate liquid measurements. So our station is prepped, labeled, and ready to go. But before we mix a single drop, we really need to pause. Handle concentrated nitric acid carefully as it is highly corrosive. Seriously, let's take a moment here. Concentrated nitric acid demands your absolute respect in the lab. It is highly, highly corrosive. We also have to be incredibly careful to avoid overheating during our next couple of steps. Always use proper safety measures, handle acids with extreme care, and respect the chemicals you're working with. Safety first, always. Okay, phase three, step-by-step procedure. Now, what's really interesting about this slide is that this precise, sequential execution is the absolute only way we're going to get a reliable, undeniable result. Let's walk right through these exact measurements. Step one, we're going to take exactly 2 milliliters of our protein solution, our egg albumin, and place it super carefully into a test tube. Step two, grab that pipette and add exactly 1 milliliter of that concentrated nitric acid right into the tube. Now, step three is where we heat the mixture. But remember our big precaution here, we need to heat this gently. Don't rush it. Step four, we wait. The suspense builds because we have to let the solution cool completely down. No shortcuts. Finally, we reach step five. This is the moment of truth. We add our alkali, the sodium hydroxide or ammonia, and we closely observe what happens. And that brings us to phase four, reading the results. This is the payoff, the exact moment where all that invisible chemistry turns into a stunning visual reveal right before our very eyes. And this brilliantly illustrates how color is the absolute main character of this scientific story. Let's break it down. When we first added the concentrated nitric acid to the aromatic amino acids and heated it, they reacted to form these yellow nitro derivatives. That is stage one. You see the formation of a clear, bright yellow color, which hints that our target molecules just might be in there. But then, when we add the alkali in stage two, oh man, the color intensifies dramatically. It makes this huge shift from yellow to a deep, vibrant orange. That orange color, appearing right after the alkali is added, is our confirmatory positive result. Seeing that yellow to orange shift officially confirms, without a doubt, the presence of aromatic amino acids, tyrosine, tryptophan, or phenylalanine right there in our sample. So let's bring it all together. The Xanthoproteic Test is just an incredibly simple, highly effective method used to detect aromatic amino acids and proteins. The whole shebang really comes down to this one undeniable, completely clear yellow to orange color shift. It's a beautifully straightforward diagnostic tool. It literally takes the invisible microscopic world and makes it vibrant, colorful, and completely understandable for us. Which brings us to the end of our investigation today, but, it leaves us with a totally fascinating question to ponder. If a simple chemical reaction, just shifting a liquid from yellow to orange, can reveal the hidden building blocks of a plain protein like egg albumin, what other hidden secrets might simple biochemical tests reveal about the complex world around us? It's like a giant puzzle just waiting to be solved. Keep questioning, keep investigating, and hey, thank you so much for joining me for this explainer.